Okay, starting off from where we were last time, this is part 2 of the video. Okay, now, your DNA is enormous. A human's DNA is like billions of base pairs together. So, in order to, it takes a few hours to do DNA replication. In order to do it efficiently, you actually have multiple spots throughout the chromosomes, all right, or the DNA when it's being replicated, where helicase is opening up, and it's opening up in both directions. And the spots where it opens up, it forms this bubble, okay, where it opens up and it looks kind of like a bubble, and we call that a replication bubble. See, it looks like a kind of like a bubble here. Helicase is opening, one helicase is opening in this direction, another one's opening this direction, and you have this bubble hundreds or thousands of times throughout your whole genome when you're replicating DNA. This part, because it's shaped like a Y, we call it the replication fork. Okay, right here, the replication fork. So the replication fork is opening, you have a replication fork over here opening in this direction, and this helicase is moving this way, so you have a replication fork opening in this direction. Okay? Okay, so helicase opened it up and formed the replication forks. Those brown things are called what? Single-stranded binding proteins. I'll try to kind of ask you, for you to quiz yourself. And what do? They prevent the two strands that we just opened from connecting back together, from hydrogen bonding back together. Okay. A replication fork is, I'll give you a second. A. All right, let's continue. All right, there are the two strands run antiparallel. Okay, it's not shown well in this simple cartoon, but the concept of antiparallel strands, the best example I can come up with would be like driving on a highway. Whatever side of the highway you're driving on, all the cars are facing in one direction. If those cars are the nucleotides, right, and they're facing one direction, that's the side of your highway is one strand of DNA. And on the opposite side of the highway, all of the cars, or nucleotides in our analogy, are facing the other direction, right? You see the tail lights ahead of you on your side of the highway, and you see the headlights on the other side of the highway. And so the cars would be facing or running in anti-parallel directions. So what we call one end of the DNA strand is the 5' end, and the other end is the 3' end. It has to do with how the carbons are counted, but we're not going to get too much into it. This little apostrophe means prime. And so if this is the 5' end on the bottom and this is the 3' end, the other strand is the opposite way, 5' and 3', okay? And the reason I'm telling you this is because it makes a difference during the speed of replication of the different strands, okay? DNA polymerase can only read in a certain direction. It can read the 3' end to the 5' end of the parent strand. Just like you can only read from left to right, you can't read from right to left or bottom to top. Polymerase can only read from 3' to 5' and can only make from 5' to 3', okay? Why does that matter? We'll see in a second. So DNA polymerase cannot begin from scratch. That's why we need a primer. So they're talking to you about the primer. What enzyme puts the primer on? The little RNA primer on? Primase. Okay, it's temporary. It's going to come off. Then DNA polymerase can do its job. it's going to read the little temporary RNA primer, all right, and the sliding clamp is going to help it stay on, and it's going to read an A, add a T. It's going to read a C on the parent strand and add a G. It's going to read a G, it's going to add a C. It's going to read a T, it's going to add an A on both strands. Notice this one is working in the direction that the replication fork's opening, and this one's working in the away from it, okay? Because polymerase can only read in one direction, and these are antiparallel. So this strand, this polymerase and this strand can be made continuously. It just keeps going in the same direction that the DNA is being opened. But this strand can only work in this direction. So it makes a little bit, and then it has to backtrack, double back over what it already made, make a little more, backtrack, make a little more. So this side is called the leading strand because it goes a lot faster. This side is called the lagging strand because it goes more slowly because it has to cover more ground. Leading strand can be made faster. Lagging strand is made in a series of chunks. Because the DNA polymerase can only read in one direction, it's on this strand, it's making DNA in the opposite direction that the fork it's working on is opening. The antiparallel nature of double-stranded DNA means that what do you think? The two strands run from 5' to 3' in opposite directions. The 5' ends of the two strands are on the opposite ends. I think it's probably A and B. The lagging strand, is it made continuously? Is it made in a series of fragments called Okazaki fragments after the scientist who found it? Is it more heavily coated with SSB proteins? Is it made more slowly in the presence of due to the presence of the sliding clamp? So it's made more slowly, but is that because of the sliding clamp? No. So the answer is B. All right, so you have to read the full question for each. Okay, then we're going to rip off the primers using what enzyme? RNase H. Okay, primase puts the primer on. RNase H removes the RNA nucleotides because we don't want them in our final DNA molecule. Okay. Then we got to fill in the gaps. Who does that? This green blob named DNA polymerase. But it can't do the last bond for some reason, the last sugar to phosphate bond. So ligase comes in and just glues the last little bonds together. Okay? RNase H does what? Removes the primers, creates the primers, breaks down the RNA, turns RNA into DNA. We know that it... Oops, I'm sorry. Oh, it degrades the RNA after the DNA template. My bad. Okay. This is pretty much just a review. I'm going to stop us probably before this finishes because I don't want to go past 10 minutes. I don't know if YouTube's going to let me upload a more than 10-minute video. So, all right, so you can review this at will. I would review it many times because I think this is, although it's a simple video, that's kind of why I like it. Okay.